Great, thank you for that. So that was a glimpse of what we think a research hospital is and what it means to us and how important it is to us at GOSH. So with that, I'm going to move to our next session, which is focused on our gene, STEM and cellular therapies theme. So if I could invite Professor David Long, who is the Deputy Theme Lead for this theme within the BRC, to join us, please, and chair the session. Thank you, David. Over to you.
Thank you, Laura. So it's a great pleasure for me to chair this session on the showcase, which is focusing on gene, STEM and cellular therapies. So we're going to have three potential talks. Well, we're going to have three talks highlighting the work of the theme over the last 5 years, and hopefully some time for questions afterwards. So our first speaker is Paul Gisson, who is the leader of the gene, STEM and cell therapy theme. And Paul's a professor in pediatric metabolic disease, and he's going to give an overview of the whole theme program. Over to you, Paul.
So you should be able to hear me now.
Yeah, all okay.
And see my talk. Okay. So thank you very much. Good afternoon. It's my pleasure to introduce the gene, STEM and cellular therapies theme and give you the overview of the activities. The aims of the GSCT theme are to build on our clinical and research expertise of developing cell and gene therapies in immunological, blood, neuromuscular and metabolic disorders. We aim to expand the range of disorders amenable to gene and cell therapies and make these treatments more effective and safer. Our theme's funding is focused on the three major areas: clinical trial support, GMP manufacturing teams, and we fund pump priming projects that accelerate preclinical development. The four-year activity overview for our theme shows that our investigators led 289 preclinical and clinical projects. 47 of these projects were phase 1 and 2 clinical trials, which recruited 85 participants. 17 were first-in-man trials, and we leveraged grant income of more than 46 million. Many of the projects led to excellent and highly impactful publications, and many of these projects were highly newsworthy, such as novel CAR T cell therapies for various disorders and revolutionary eye gene therapies, as you could see here. The GMP facility is the largest academic facility in the UK with 7 suites. 98 products were produced for academic and industry-sponsored trials. Patients were treated in the UK and also internationally. And finally, last year, a collaborative agreement was signed between GOSH and Viracell in order to increase the productivity of the facility further and facilitate gene therapy vector production. 2 years ago, the GSCT researchers moved into the Zayed Center for Research for Rare Diseases in Children.
The laboratories here are really set up for advanced therapy medicines. So that means medicines which involve cells or gene manipulation. Really, this facility provides us with increased capacity to make more different medicines for patients at Great Ormond Street Hospital.
Working in the Zayed Center has changed the way that we work. I think having this co-localization of research teams that develop the therapies and clinical teams that deliver the therapies has been beneficial for everybody. So the research scientists developing these treatments can really see the impact of their work, and the clinical teams can understand how the treatments have come to be in the clinic. Particularly here in the Zayed Center, with the outpatient facilities and the patients coming in, the researchers get to see the patients and really understand the impact that their work has had on those patients and families. And then the patients and families can also see how much work is ongoing here to develop new cures for other diseases.
We couldn't tell you about all the research projects that are going on within the GSCT. The research focus for the theme lies in 3 different areas at least. And these are hematopoietic stem cell and gene therapies. And you will hear further from Claire and Georgia after my talk. The next topic is the engineered T-cell therapies, and I will mention the work of Karin Strathoff and also Sarah Garashian on CAR T-cells in their disorders, and also finally, the AAV gene therapy, and I will talk about the collaboration with the Sydney groups on developing and testing novel organ targeting vectors. Dr. Sarah Garashian leads a phase 1/2 clinical trial in patients with acute lymphoblastic leukemia using low affinity chimeric antigen receptor T cells. The researchers hypothesized that lowering the affinity of CARs will be advantageous. Indeed, they found that the novel therapy was equivalent in efficacy to licensed treatment, but had better toxicity profile, and it is now going through licensing for the adult patients. Neuroblastoma is a cancer of central nervous system. Some patients have a particularly poor prognosis. Karen Strathoff showed that GD2 surface glycoprotein can be safely targeted with CAR T cells in a phase 1 study. And the signal of activity reduction in tumor burden in bone marrow and soft tissue sites of disease, as shown here, can be achieved with CAR T cells. Some patients on this clinical study had benefit, no longer requiring analgesia, improved performance score, but this benefit was short-lived. Karen now works on applying advanced T-cell engineering approaches to achieve CAR T-cell persistence in order to achieve more durable clinical response. Wellcome Trust Career Development Fellow Alexandra Cranes is developing cell therapy for patients born without a thymus. This is a cross-theme BRC collaboration. And what Alex is doing, she's optimizing method of differentiating thymic cells from patient-derived induced pluripotent stem cells to start with, and then eventually, in order to use these cells for treating patients without the thymus. We developed a close collaboration with the scientists at CMRI, the Children's Medical Research Institute, on developing adeno-associated viral vectors gene therapies. 2 PhD students, which are joint between Gosh and Sydney, were able to make breakthrough in the understanding of how the human liver cells are targeted by the AAV and develop new highly efficient capsids. In a translational collaboration, one of the novel capsids is used by the researchers at Gosh in a first-in-human trial in a severe metabolic disorder, OTC deficiency. In a collaboration with the Novel Therapies theme of the BRC and UCL School of Pharmacy, we opened an AAV viral synthesis facility. Julia Massaro, the facility manager, has already been involved in more than 20 projects with various groups, and the aim of these projects is to accelerate clinical trial development. And that's a remarkable achievement considering that this facility only opened in November 2020. After the initial successes with Orchard and Autolus, the third spin-out company with the GSCT investigator involvement was formed in 2021, and more are in progress. The aim of this work is to accelerate access to the cell and gene therapies for the patients. Hot off the press, and only from last Friday, one of the spin-outs, Orchard Therapeutics, announced NICE approval for reimbursement for hematopoietic stem cell gene therapy for a rare metabolic neurodegenerative disorder, metachromatic leukodystrophy, for patients in the UK. Through world-leading excellence in clinical research, we attract industry collaborations. More than 13 million investment into research was brought in by GSCT investigators in the last 4 years. And finally, our investigators do a lot of work in disseminating the knowledge about gene, stem, and cellular therapies. Scientists organize webinars for the families, the work with the Young Patient Advisory Group at Gosh to create accessible materials, get involved in Royal Institution Christmas lectures, and more traditionally, share the results of our work with colleagues at various conferences. Thank you for listening, and I pass you over back to David. There.
So thank you, Paul. That was a great overview. So our next speaker is Claire Booth, who is a consultant paediatric immunologist and also a professor of gene therapy and molecular immunology. And Claire is also deputy lead of the Gene, STEM and Cellular Therapies team. And she's going to talk about gene therapy for ADA SCID.
Great. Can you see my slides okay?
Yeah, that's fine.
Okay, great. So thank you for inviting me today to share one of our more successful research stories, gene therapy for ADA SCID, which has really transformed the lives of so many patients and families. I'm sorry. It's not moving along. There we go. Okay, so the gene therapy team at Great Ormond Street Hospital and the Institute of Child Health, founded by Adrian Thrasher and Bobby Gaspar over 20 years ago now, has been pioneering gene therapy treatments, offering patients potential cures for often fatal immune deficiency disorders. And the first trials were undertaken for severe combined immune deficiency, or SCID, where children are born without a working immune system due to genetic mutations. And the successes that we had in these diseases means that the technology has been applied to a wide range of other genetic conditions. And here at GOSH, we've treated over 120 patients for 15 different conditions. And this is a photo of Reese, who was the first patient treated with gene therapy for SCID here at GOSH 21 years ago last July. And this signifies a huge achievement for the field. And it's really patients like Reese and his family who are the true pioneers. And Reese is still doing very well, living a normal life and is infection-free. So ADA SCID, so adenosine deaminase deficiency, it's a disorder of pure metabolism, and the enzyme ADA converts a compound called deoxyadenosine to deoxyadenosine. In the absence of that enzyme, an alternative pathway is used, which leads to the accumulation of toxic metabolites, primarily something called DATP, which is toxic to many tissues, but it has a really profound effect on the lymphocyte development and function. And as such, patients present with SCID, so severe combined immune deficiency, which is invariably fatal in the first year of life without any treatment. So in addition to the profound panlymphopenia that we see, which affects T, B, and NK cells, patients manifest a range of systemic clinical features, including developmental disorders, deafness, lung problems, urogenital problems, skin problems, and bone lesions as well. And although definitive treatments can cure the immune deficiencies and the immunological aspects, many of these systemic features remain. So following diagnosis, supportive care is the mainstay of treatment until a definitive treatment options available. And this includes antibiotic prophylaxis, preventative medicines, replacement immunoglobulin therapy, so monthly infusions, and enzyme replacement therapy with something called PEG-ADA. And this is a weekly intramuscular injection, which does allow some often transient improvement in the immune system, but its effect wanes over time, and it's extremely expensive and not universally available. So in terms of definitive treatment, allogeneic hematopoietic stem cell transplant or bone marrow transplant is curative, but risk complications of poor metabolic and immune recovery in the setting of an unconditioned infusion, and the risks of alloreactivity and graft versus host disease, particularly in the context of mismatched donors. And so this survival curve here shows that that survival following transplant from non-HLA identical related donors can fall below 50%. And it was for this reason, really, that ADA SCID was one of the first diseases tackled with gene therapy over 20 years ago. And initial trials using a gamma-retroviral vectors developed here led to the approval in Europe of a gamma-retroviral gene therapy called Strimvelis, which is available in Milan as a fresh drug product. But due to safety concerns over gamma-retroviral gene therapy and several other genetic conditions where patients treated with this type of vector develop leukemia as a result of the vector, the field has moved towards using something called a lentiviral vector, which to date has a much improved safety profile. And more recently, our team has published more long-term follow-up transplant data on this condition, again supporting the use of gene therapy as a treatment for this condition with really favorable long-term outcomes. So so this is a simplified graphic that highlights the main steps of gene therapy, starting with the harvest of bone marrow stem cells or hematopoietic stem cells, which we collect either through bone marrow harvest or via leukapheresis of mobilized peripheral HSCs. And then the progenitor cells, the CD34 cells, are purified and cultured with cytokines and incubated with the lentivirus, which is a virus that we use to carry a working copy of the gene into the patient's stem cells. This is the self-inactivating lentiviral vector that was developed here with the human ADA transgene expression driven by the mammalian EFS promoter. And once a sufficient number of CD34 cells was confirmed to take forward to transduction, patients received a single dose of low-dose busulfan conditioning and then a 24-hour washout period before infusion of gene-corrected cells. Now, BRC funding supports vital steps in this process. It funds members of our manufacture team who actually manipulate the cells and make the drug product, and our specialist nurses who are involved at every step of the patient journey, including infusing all of our gene therapy products. The BRC also supports members of the wider team. So a lot of people are involved in translating our research to patients and delivering successful clinical trials such as this. And this is a snapshot of our team before the pandemic. And the BRC supports our nurses, pathway coordinators, project managers, manufacture team, QA team, and contracts team. So really the whole infrastructure of our service. So last year, we published in the New England Journal of Medicine the results of our collaborative trial with UCLA, where we had generated compelling evidence that this treatment works across what is a very large number of patients for an ultra-rare disease. So we showed here an integrated analysis of two prospective, non-randomized early phase clinical studies conducted in the US using both a fresh and frozen or cryopreserved drug formulation, and then a separate analysis of a study undertaken at Great Ormond Street Hospital. We have an ongoing trial underway in the UK using a frozen drug product, and although close to recruitment, follow-ups ongoing. But altogether, we presented data on 50 patients treated across studies, so 30 in the US and 20 in the UK. And the inclusion criteria listed here is for the UK study, but it was similar for the US cohort. Patients were eligible if they lacked an HLA match-related donor for transplant. And in the UK, we were able to treat older patients if they were eligible, if they had evidence of preserved thymic function. So safety endpoints were incidence and grade of adverse events and serious adverse events, including detection of clonal expansion. Primary efficacy endpoints were overall survival and event-free survival at 12 months, which meant that patients didn't have to go back onto enzyme replacement therapy or progress to a rescue bone marrow transplant. We also looked at engraftment of gene-corrected cells, immune recovery and metabolic detoxification. And then secondary endpoints included overall survival and event-free survival at 2 years in the US study and 3 years in the UK study, alongside the parameters listed here, and also rates of severe and opportunistic infections requiring hospitalization. So I'm going to use this slide not only to show you 100% survival across all groups with 3 years follow-up in the UK and 24 months follow-up in the US, but also to orientate you to the following slides where the UK study group is represented in green and the US study group in blue. So event-free survival was 97% in the US cohort at 2 years and 95% in the UK cohort at 2 years and 3 years. One patient in the US study was withdrawn after 6 months because of the lack of engraftment of gene-corrected cells and went back onto enzyme and had a bone marrow transplant. And one patient in the UK started enzyme replacement again 12 months after treatment due to insufficient engraftment and low ADA activity levels. So when it comes to measuring ADA activity, which is the enzyme that we've replaced, both sites use slightly different assays, so results are shown in separate graphs, but what you can appreciate is a very rapid rise in ADA activity in the first 3 months after treatment, and these levels remained within or above those seen in healthy children all throughout follow-up. And looking at immune recovery, we saw really consistent and robust lymphocyte or T cell recovery with participants achieving or coming close to achieving T cell counts within the normal range for their age. And this was true for both CD4 and CD8 populations. And looking at humoral recovery and reflecting normal B cell development now, the majority of patients had increased IgM and IgA levels to within normal limits, but really importantly, sustained normal IgG levels after stopping immunoglobulin replacement, so they could stop their monthly infusions. And for the patients who've been vaccinated after stopping immunoglobulin, we could show normal antibody responses to tetanus vaccines. So in summary, we were able to show that using our lentiviral gene therapy across 3 different trials, encompassing 50 patients, that we could really show 100% survival and 95% event-free survival at the last data analysis, and really robust immune recovery and sustained metabolic detoxification, but also importantly, a very favorable safety profile. So the fantastic results we published attracted a lot of press attention, and our patients who have been treated here were happy to share their stories about exactly how transformative this treatment had been for them and promote awareness of rare diseases and gene therapy trials. And this is Sarah, who's now 5, and she can lead a normal life. She can go to school, she can have birthday parties, go on holidays, all because of the treatment that she received here. So there's well over 100 people to be thanked and acknowledged who supported this program, not including the patients and families. And none of this would have been possible without the amazing team that we have here. Thank you.
Well, thank you, Claire. That's a really fantastic example of the impact that the work of the theme is having. And so our last talk is from Giorgia Santilli, who's an associate professor and a member of the BRC junior faculty. And so she's going to give a brief talk about genome editing in hematopoietic stem cells for the treatment of primary immunodeficiency. Okay, so over to you, Giorgia.
Thank you, David. Let me just share. Can you see my slides?
Yes, we can see them.
Okay, thanks. All right, thank you, and good afternoon, everyone. Yeah, I've just prepared a few slides to show you where we are with the genome editing in hematopoietic stem cell for the treatment of primary immunodeficiencies. And Claire has talked about a conventional gene therapy approach for ADA-SCID, where we deliver the therapeutic gene with the lentiviral vector. Here I'm going to talk about a different approach, where we are trying to target the insertion of the gene in a desired locus by using a Cas9 endonuclease that creates a double-strand break in the DNA, and by harnessing the homologous recombination machinery of the cells to repair this break with a donor template. And if we deliver the donor template with a non-viral methodology or with viruses that do not integrate in the genome, then we can achieve seamless correction. And if we knock in the therapeutic gene next to its own regulatory sequence, then we can also achieve physiological expression of that gene. We've been optimizing this for a number of primary immunodeficiency over the years, among those X-linked agammaglobulinemia, which is an antibody disorder and is caused by mutation in the gene encoding Bruton tyrosine kinase. And this protein is important in the development of B cells in the bone marrow. So patients who lack BTK have absent B cells and no immunoglobulins, and this leads to severe and life-threatening infections that are ameliorated by immunoglobulin replacement therapy, which is a lifelong treatment and also very expensive for NHS. So what we are proposing here is a potential cure, a one-off treatment that can also address the non-humoral component in XLA, because it's becoming increasingly clear that BTK is also expressed and has a role in other lineages. For example, it's expressed in myeloid cells and has a role in inflammation. We also need to restore a physiological pattern of expression of BTK, because if BTK is always turned on, that can cause cancer. And other strategies, for example, gene editing strategies or base editing strategies, where we're trying to correct the single mutation, will not really work for XLA, like for a number of other primary immunodeficiencies, where mutations are actually scattered throughout the locus. So we have optimized a protocol where to nucleofect a Cas9 ribonucleoprotein complex targeting an early exon of BTK, and then we deliver the donor template with an adeno-associated virus 6, which hardly ever integrates into the cell. And we have been doing this with money from the BRC. And as you can see here in this figure, we have tested the protocol in hematopoietic stem cell from different donors, and we could achieve more than 20% correction, so targeted integration, with an average of 40% among 5 donors. And if you look at the Western blot here, you can see that with our gene editing, we can restore levels of BTK which are similar to a wild type. And then we spent some time in the lab trying to perfect a differentiation protocol to obtain immature B cells starting from hematopoietic stem cell. And we could reproduce the defects observed in XLA. And in fact, if you look at this little square here, at week 4, we do have pre-B cells starting from hematopoietic stem cell from healthy donors. And these pre-B cells are able to progress to maturation to immature B cells and express IgM, while this does not happen for the XLA sample. And gene editing can rescue this phenotype. So we have now concluded the proof of concept, and we are moving on to safety studies. We have also noticed that there is a problem of toxicity in some donors more than the others, and we are trying to understand why is that. And also there is a bit of variability between donors. And so although this may not be very important for diseases like XLA, where there is a strong selective advantage of corrected cells, so we can get away with a low level of correction, it could be crucial for other primary immunodeficiencies. And with the money, again, from BRC, we are now trying to optimize new donor templates. So we are screening novel AAV serotypes, for example, but also trying to use a non-viral RNA donors. And I'm not going into technicalities here, and I'm just going to finish off thanking everyone at MCI involved in this work. So this is a very collaborative work. And thanking my team, in particular Samir Bahal and Thomas Whittaker, who are working on different aspects of this project. And also I want to thank the GMP manufacturer team, who has been instrumental in bringing to the clinic several proof of concept that we have developed, and who I hope that they're going to help us again scaling up this process and moving into the clinic this protocol for XLA in the next 4 to 5 years. Thank you.
Thank you, Georgia. That was great. It's a very nice example of how a small amount of money from the BRC is really developing into a much larger project. I think we have one question from Laura.
We do. There's one question on Slido, and it says that gene therapies offer great opportunities, but they are very expensive. Do you think the NHS will be able to afford to offer them more widely? That could be to any one of the speakers.
Okay, Paul or Claire, do you want to do that one?
I can start and Claire could finish. I don't know if you see me. I hope you do. So this is a very, the cost is always a very emotive topic. Of course, gene therapies per patient are very expensive. However, well, first of all, it's we're talking about rare diseases. And when you only have 5, 10, 20 patients, there is still an expenditure from the company developing therapy that somehow needs to be got back. So if you compare, if the expenditure is similar on developing therapy in a drug for hypertension, and then you can give it to thousands and thousands and hundreds of thousands of patients, of course you can afford to cost it a lot less. And when you have only rare disease, a few patients, and that's why it costs more. But in fact, the government will spend a lot more on cheaper options. So that's one point. Two is actually, gene therapies on the whole are one-off treatments, and you spend it once, and then provided that this is efficacious and safe therapy, then it's close to a cure. I'm always worried of saying that it's definite cure. For example, if we're giving enzyme replacement therapy, then it's a lot more expensive because you have to spend more or less the same amount of money every year for life versus once off. So I think these are the 2 points I wanted to make. Claire.
And thanks, Paul. I think that's absolutely right, that for some of these conditions, the standard of care treatments is extremely expensive. And actually in those situations, gene therapy can offer a relatively cost-effective opportunity. I mean, the other thing to say is that some of the conditions that we're tackling with gene therapy don't have any other treatment option. So it really is the only option for these patients. But those of us working in the gene therapy field, here at UCL, GOSH, the BRC, and with our European partners are really working towards improving the accessibility of these treatments, but also trying to understand where there are points in the manufacture process where we can save money, how we can do things more cost effectively. And so some of those projects are part of our research theme as well.
Perfect, thanks both. I think we've run out of time now, so I'm going to hand back to Laura, but just thank you for everybody who's spoken in this session. It's a really good, nice overview of the theme. Over to you, Laura.
That's great. Thank you very much, David. That's the end of the first half of our showcase. We're delighted that there are more than 200 people online enjoying hearing about some of the research successes of the GOSH BRC. We're going to take a short break now and we'll be back at 3:30. During the break, can I remind you, that the image competition voting is well underway now online. There are lots of people voting. And also that we're going to play some slides during the break, which are just there for you to view, that highlight some more of the successes from the GOSH BRC. So see you shortly. Thank you.